Our final presentation of today's morning session is by Naveen Kumar, and the presentation is entitled Understanding and Enhancing Drought Tolerance in Virginia Type Peanut.
Thank you for the brief introduction. Good afternoon, everyone. Today I'm going to deliver my talk on understanding and enhancing drought tolerance in Virginia type peanut. Here is the outline for my presentation. Firstly, I want to justify my research work, why we are studying drought in Virginia, Carolina region. Second part will focus on understanding drought physiology and genetics in our germplasm with the objectives framed in our study to enhance drought tolerance in Virginia type peanuts, followed by material and methods, results, and future plans. Why drought in VC region? Here are a few factors that drive us to study drought in VC region. As we all know, to grow and eat profitably, peanut require a uniform distribution of water ranging from 1 to 2 inches per week depending upon the different growth stages. And this graph shows the pattern of rainfall in Virginia, Carolina region in last 2 decades. On the x-axis, you will see the weeks of the growing season starting from May, assuming as our plantation date. And on the y-axis, you can see the amount of rainfall received in inches. These vertical bars dividing the growing season into 4 different growth stages of peanut, beginning from flowering to physiological maturity. This concentric circle reflects the amount of rainfall, average rainfall received in that particular week during 2 decades. These solid bars, horizontal solid bar, reflects the average amount of weekly rainfall received during these growth stages in the VC region. And we can clearly notice from this graph that the VC climate provide an appropriate rainfall or precipitation to the peanut in the beginning of the season, but later part of the season, the amount of rainfall deficits, which lead to yield penalty to the peanut farmers. And the second factor is the sandy soil. It is like a double-edged sword because peanut prefers to grow in sandy soil, as sandy soil is favorable for pot and pegging. But sandy soil has very low water holding capacity that makes peanut more prone to a drought. Irrigation can be supplemented under drought events, but 85% of the peanut acreage in VC region is under rain-fed conditions. And one of the report shown by Dr. Balota shows that drought caused an economic loss of $50 million to the peanut farmers in this region. So by considering all these factors, the most appropriate solution to mitigate drought is to develop drought-tolerant cultivars. And to achieve this broad objective, we have hypothesized that sufficient genetic variation for different physiological traits which contribute for drought exist in our germplasm. But for precise identification and validation of those traits which contribute to drought is done by phenotyping and genotyping of recombinant inbred line for drought tolerance. Now moving towards the material and methods we used in our study. For phenotyping and genotyping, we use 3 recombinant inbred populations for our study material. These parents are the advanced breeding lines which were selected from the previous study based upon the phenotypic differences on the drought and yield. For example, the parent NO506, Phillips, and ICG are drought resistant, whereas NO4074CT is drought susceptible. And we used the experiment design RCBD with two replications. And these experiment is put in 2 different locations, one at North Carolina at Lewiston and second at Suffolk in Virginia. And Today's talk, I will focus on population 3. Now moving toward the phenotyping. So the population 3 was phenotyped by using NDVI and CTD at different growth stages and at different drought severity in 2019. NDVI is calculated by measuring the wavelength within the near infrared and red region of the electromagnetic spectrum. NDVI relates to the chlorophyll content based upon its absorption property. NDVI is also a significant indicator of plant health, and it is effectively used to determine the yield and biomass under stress. The value of NDVI lies between 0 to 1 for the plants. And the more the NDVI value, more is the plant healthier, as you can see in the picture below. We use handheld green seeker to measure the NDVI for this population. Similarly, CT is also used as a selection tool for hot and drought tolerant genotypes. CTD is measured with the difference in the canopy temperature and the ambient temperature. CTD work basically on the principle when the plant are under stress, transpiration reduces because of stomatal closure, but the drought tolerant genotypes or plants continue to transpire and the canopies are cooler. So the negative value shows cool canopies, which reflects drought tolerant plants or cultivar, whereas positive CTD values reflects hot canopy and drought susceptible. And we measure the CTD with handheld infrared thermometer of agritherm. Now after phenotyping, we genotype all the three population. We will genotype all the three population using genotyping by sequencing method. The GBS approach is followed as shown in the flow. We have already extracted DNA across all the three populations. The DNA templates are used to construct the GBS libraries, first pooling the DNA and then ligating with the optimized adapters and then followed by barcoded. And once the GBS libraries will be constructed, they are sent for sequencing, and we use Nova Sequencing 6000 as our sequencing platform. And this sequencing data is further analyzed to identify the genetic markers which are associated for drought in our germplasm. So after genotyping, I will like to discuss the results in population 3. In population 3, first we perform descriptive statistics on the data to get summary statistics on all the measurements, including NDVI and CTD. Then we analyze the phenotyping data using the principal component analysis method to reduce the large number of variables in the data and extracting the important one depending upon the magnitude and direction of the variables. In this graph, the PCA report shows that the first principal component analysis account about 60% of the variation in our total data, and together both the components contribute about 25% of the variation in our data. Based upon this analysis, the pools, we made two pools, high and low, which contribute about 15% of the total population entries from population 3. And I will show in the coming slides how we select these entries on the basis of NDVI and CTD. So in this graph, on the x-axis, you see the NDVI, on the y-axis, you see the entries. And these different colors shows the pool. So the green color will show the high pool entries and the red will show the low pool entries. And as I earlier mentioned, the value of NDVI lies from 0 to 1. More the value of NDVI, more is the healthier plant. And you can see that in around 0.75, which around reaches to 1, you can see the more green dots, which represent the entries which we selected for high pool. And similarly, opposite of that, The green dots represent the entries which fall under low pool. Similarly, for the CTD, as the value of CTD lies between minus to +1, and the minus values represent cooler canopies that the plants perform better in water stress. So same color code. So the green dots shows the number of entries which perform better and have negative CTD values as compared to the red. And we pool these green and red into both high and low pool. So moving towards what we have concluded from population 3 based upon the PCA analysis, that we have selected around 26 entries for each pool designated as high and low. And these entries are now put further for detailed phenotyping in the Rainout shelter with more data collection on the visual ratings like wilting, carbon dioxide assimilation, chlorophyll fluorescence, and disease rating. So what are the future plans we have to accomplish this project? The phenotyping would be continued in 2001 with the additional data. And once the genotyping is done, we will identify the genomic markers associated for drought in our germplasm. And these selected lines, which are under testing, would be used to validate the panel of markers we identified through phenotyping and genotyping. Finally, I would like to thank my committee member and major advisor, Dr. Maria Balota, my co-advisor, David Haig, who is helping me with the genomic part, Dr. Jeffrey Dunn, who is helping me in establishing the experiments at Lewiston in North Carolina, all the lab members who helped me in setting the trials and collection of data, and finally the funding agency, NIFA, who fund this project. Thank you for your kind patience.
Thank you. Thank you for the presentation. We have time for questions. First question is, again, from Dr. Barry Tillman. What underlying traits do you think might explain cooler canopy temperatures or other indicators of drought tolerance?
Sorry, can you say again?
Yes. The question is, what underlying traits do you think might explain cooler canopy temperatures or other indicators of drought tolerance?
So there might be different traits depending upon the architect or architect of the lines, because this population is constructed from a Spanish type and Virginia type parents.
Okay. Looks like there's no further questions from the audience. I want to thank you again for your presentation.
Thank you.